Welcome, dear students. Today, your dedicated participation is crucial, as understanding stem cells not only deepens scientific knowledge but also equips you to critically engage with the cutting-edge innovations and ethical challenges that define modern biomedical research, shaping the future of healthcare system. Today's learning objectives are: critically evaluate the biological properties, classification and potency of stem cells. Investigate the mechanisms of adult stem cell mediated tissue maintenance. Master the properties, culture techniques and applications of embryonic stem cells and ethics. Explore the principles and techniques of therapeutic cloning, that is SCNT. We'll later on integrate knowledge of adult stem cells, embryonic stem cells and innovations with therapeutic cloning. Now, moving on to introduction to stem cells. Stem cells are unique cells defined by their dual capacity for self-renewal and differentiation into specialized cell types. They play central roles not only during embryogenesis but also in maintaining and repairing adult tissues throughout life. Broadly, they are categorized into embryonic stem cells, which are pluripotent, and adult stem cells, which are multipotent or tissue potent. Moving on to key properties of stem cells. Number one, self-renewal. The ability to undergo multiple cycles of division without losing stemness. Potency. The capacity to differentiate into specialized cell types. Embryonic stem cells are pluripotent, whereas adult stem cells are multipotent. Moving on to the introduction to stem cells in modern biology. Stem cells form the cornerstone of regenerative medicine, bridging developmental biology and clinical innovation. Their history spans from the discovery of hematopoietic stem cells by Till and Mikolu in 1961 to Martin Evans' isolation of embryonic stem cells in 1981 and Shinya Yamanaka's breakthrough of induced pluripotent stem cells or iPSCs in 2006. These advances, honored with Nobel Prizes, highlight the transformative impact of stem cell biology in disease modeling, tissue repair, and personalized therapies. Dear students, moving on to classification and potency. Stem cells are classified as totipotent, that is zygote, pluripotent, the induced pluripotent cells, multipotent, adult stem cells like mesenchymal stem cells, and unipotent like spermatogonial stem cells. Embryonic stem cells and therapeutic cloning offer broad regenerative potential but raise ethical concerns, while adult stem cells minimize rejection risks. Making potency distinctions are key to ethical debates and applications, as multipotent cells minimize rejection risks compared to pluripotent ones. Embryonic stem cells derived from blastocysts exhibit pluripotency, while therapeutic cloning somatic cell nuclear transfer generates patient-specific clones for therapy. Dear students, moving on to maintenance of adult tissues by these stem cells. Adult stem cells or somatic stem cells are multipotent cells residing in specific niches that maintain tissue homeostasis by self-renewing and differentiating into tissue-specific lineages, like hematopoietic stem cells found in the bone marrow. They maintain lifelong production of blood cells, that is erythrocytes, leukocytes and platelets. They are controlled by niche signals, cytokines and transcription factors, for example GATA2 to PU.1. Clinical relevance: bone marrow transplantation in leukemia and aplastic anemia. Epithelial stem cells: they are found in basal layer of the skin and intestinal crypts. In the intestine, crypt-based columnar stem cells give rise to absorptive enterocytes, goblet cells, Paneth cells and enteroendocrine cells. Rapid turnover ensures continuous epithelial barrier function. Neural stem cells reside in the subventricular zone, or SVZ, and hippocampal dentate gyrus. They differentiate into neurons, astrocytes, and oligodendrocytes, which are involved in plasticity, learning, and limited repair following injury. Then moving on to mesenchymal stem cells, or MSCs. They are found in bone marrow, adipose tissue, and umbilical cord. They differentiate into bone, cartilage, muscle, and fat, and they act as immunomodulators in inflammatory diseases. Muscle satellite cells. Stem cells in skeletal muscle located beneath the basal lamina. Key for muscle repair following injury. Controlled by transcription factors such as PAX7 and MYOD or MyoD. Mechanisms of tissue maintenance. Adult stem cells maintain tissue through asymmetric division, producing one stem cell and one progenitor, preserving the pool while generating differentiated cells. In the hematopoietic system, HSCs divide asymmetrically, yielding progenitors for erythrocytes and leukocytes, regulated by transcription factor 1 or TCF1 and polycomb repressive complex 1 or PRC1. Intestinal stem cells renew epithelium every 4 or 5 days via WNT and Notch with Equitate skewed family BHLH transcription factor 1 or ASCL1 driving differentiation. PG research uses lineage tracing in mice to track stem cell contributions, revealing exhaustion in aging due to niche dysfunction. Moving on to niche regulation and signaling pathways. Stem cell niches provide soluble factors, for example, stem cell factor for HSCs and cell-to-cell contacts, for example, cadherins, to control quiescence and activation. In neural stem cells or NSCs of the subventricular zone, the sonic hedgehog, SHH, and fibroblast growth factor 2, FGF2, promote neurogenesis, while aging impairs niche vascularization. Challenges in adult stem cell research. Adult stem cells' limited potency and niche dependence poses challenges for expansion and therapy. Heterogeneity with quiescent versus active subpopulations complicates isolation. Single-cell RNA sequencing reveals transcriptomic diversity in HSCs. Moving on to embryonic stem cells, their properties and applications. Origin and pluripotency. Embryonic stem cells are derived from the inner cell mass of the blastocyst and are pluripotent, meaning they can differentiate into all three germ layers, the ectoderm, mesoderm, and endoderm. First isolated from human blastocysts in 1998 by James Thompson. Embryonic stem cells express core pluripotency markers such as OCT4, SOX2, and NANOG. They demonstrate developmental potential by forming teratomass in mice, making them invaluable for studying early development and disease. Dear students, let's talk about properties of ESCs. Pluripotency ability to generate diverse cell types. Unlimited proliferation under defined culture conditions. Genome editable using CRISPR-Cas9 enabling disease modeling, and culture and maintenance of embryonic stem cells. Embryonic stem cells are cultured on feeder layers or matrices like matricial with growth factors such as basic FGF to sustain pluripotency. Differentiation is directed by specific cues such as retinoic acid for neural induction. Modern protocols employ feeder-free systems and ROCK inhibitors, improving survival and scalability for regenerative applications. Dear students, moving on to applications of ESCs in research and therapy. Number one is regenerative medicine. Differentiation into cardiomyocytes. talking about heart failure, dopaminergic neurons in Parkinson's disease or pancreatic beta cells in diabetes, and retinal pigment epithelium, macular degeneration. Drug discovery and toxicology testing. Embryonic stem cells derived cells provide in vitro platforms, for example, cardiotoxicity testing. Disease modeling. Engineered embryonic stem cells carrying patient-specific mutations allow study of mechanisms in genetic disorders. Later, ethical considerations and limitations. The derivation of ESCs involves embryo destruction, raising significant ethical debates. Additional challenges include tumor genicity from residual undifferentiated cells and potential immune rejection. Alternatives like induced pluripotent stem cells and advanced differentiation protocols are being developed to address these issues. Dear students, let's talk about therapeutic cloning, the concepts and the techniques. Therapeutic cloning or somatic cell nuclear transfer is a technique where the nucleus of a somatic cell is transferred into a enucleated oocyte to create patient-specific embryonic stem cells. Therapeutic cloning or somatic cell nuclear transfer generates patient-specific embryonic stem cells for regenerative therapy, avoiding immune rejection by using autologous cells. Developed from Dolly the sheep's cloning in 1996 by Ian Wilmette, it reprograms adult nuclei to totipotency using enucleated oocytes. For PG students, SCNT's rationale lies in personalized medicine, treating conditions like spinal cord injury with patient-derived neural cells. The techniques of SCNT are vital steps in SCNT. Isolation of donor nucleus from somatic cell, then enucleation of recipient oocyte, transfer of nucleus into enucleated oocyte, activation of oocyte, and blastocyst formation. Later on, harvesting of these embryonic stem cells from inner cell mass. Moving on to applications of SCNT. They are patient-specific stem cells, avoids immune rejection in transplantation. Disease modeling. Embryonic stem cells carrying patient's genetic material allows study of pathogenesis. Regenerative therapy. Development of genetically matched tissues for repair. Examples of research. Generation of human embryonic stem cells from cloned blastocyst, first reported in 2013. Progress in modeling mitochondrial diseases, neurodegenerative disorders. Moving on to challenges and ethical concerns. Low efficiency and high rate of abnormalities. There's a requirement of donor oocytes, which raises ethical and logistical issues. Risk of reproductive cloning if misused. Comparative insights. Adult stem cells versus embryonic stem cells and therapeutic cloning. Potency. Adult stem cells are multipotent, meaning they can differentiate only into limited range of cell types related to their tissue of origin, whereas embryonic stem cells are pluripotent, capable of giving rise to almost all cell types in the body. Therapeutic cloning-derived embryonic stem cells are also pluripotent, but with the added advantage of being patient-specific, reducing the risk of immune rejection. Adult stem cells are found in tissue-specific niches such as bone marrow, skin and intestinal crypts. Embryonic stem cells are obtained from inner cell mass of blastocyst. Therapeutic cloning-derived embryonic stem cells are produced by somatic cell nuclear transfer, in which a patient's somatic cell nucleus is transferred into an enucleated oocyte. Moving on to clinical use. Adult stem cells already have established applications, with bone marrow transplantation being a prime example in clinical practice. Embryonic stem cells are still in the trial stage, being explored for regenerative therapies, disease modeling, and drug testing. Therapeutic cloning-derived embryonic stem cells remain largely experimental, though they hold significant promise for personalized medicine and cell replacement therapies. Ethical concerns. Adult stem cells raise minimal ethical concerns since their collection does not involve embryo destruction. Embryonic stem cells raise high ethical concerns because obtaining them requires the destruction of human embryos. Similarly, therapeutic cloning-derived embryonic stem cells face significant ethical issues as their production involves the use of human oocyte and poses the risk of misuse in reproductive cloning. Moving on to clinical implications and future prospects. Personalized regenerative therapy. Future regenerative medicine will increasingly rely on patient-specific stem cells, either derived through somatic cell nuclear transfer or induced pluripotent stem cells. These cells minimize immune rejection risks and open avenues for individualized treatments. Coupled with CRISPR-Cas9 genome editing, mutations can be corrected before clinical applications, offering curative strategies for genetic diseases such as sickle cell anemia or cystic fibrosis. Now, integrating stem cells with biomaterials and organoids. The combination of stem cell technology with biomaterials, scaffolds, and organoid models is revolutionizing tissue engineering. Engineered organoids derived from embryonic stem cells or induced pluripotent stem cells simulate organ functions, providing platform for drug testing and transplantation. Long-term prospects include whole organ regeneration, potentially addressing the shortage of donor organs. Ethical considerations. Embryonic stem cell research faces ethical challenges due to destruction of embryos traditionally guided by the 14-day rule. Therapeutic cloning bypasses embryo use but introduces concerns about oocyte donation and exploitation, such as treatments for spinal cord injuries or degenerative diseases against the moral status of embryos within frameworks like Oviedo Convention. Moving on to regulatory frameworks. Regulations vary across countries. In the US, NIH guidelines restrict federal funding for ESC lines, while in UK, the Human Fertilization and Embryology Authority, HFEA, licenses therapeutic cloning. For postgraduate scholars, understanding these differences is crucial in navigating clinical trials, approvals, and ensuring safety in stem cell-based therapies. Moving on to future directions and innovations. Future stem cell therapies integrate CRISPR-Cas9 genome editing with SCNT to produce gene-corrected embryonic stem cells targeting genetic disorders like cystic fibrosis. Personalized regenerative therapies leverage patient-specific iPSCs or SCNTs derived cells to minimize immune rejection. Combining stem cells with biomaterials and organoids enable organ regeneration, such as bioengineered livers. Postgraduate innovations explore organ-on-chip platforms for drug testing, enhancing precision medicine and xenotransplantation, using cloned porcine organs for heart transplants, addressing organ shortages. These advancements require robust ethical and regulatory frameworks to balance innovation with societal values, to drive transformative solutions in regenerative medicine and tissue engineering. Together, these advances promise a new era of personalized, ethically responsible regenerative medicine. Dear students, as we conclude, remember that mastering the complexities of stem cell research stands at the heart of biomedical innovation, opening pathways to regenerative therapies, organ engineering, and gene-edited innovations like CRISPR-edited organoids, cures for once incurable diseases and to transform the future of medicines. Thank you, students.